Howdy, guys. I seem to have jumped right in. Right in. I skipped my intro, my other intro with my science of doot, 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 doot. Doot, 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 doot. Doot, 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 doot. Doot, 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 doot. Doot, 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 doot. So that was my intro. I seem to skip that one, but that's okay. Hello. How are you guys doing? I have a new co-host. I have a new co-host for my show, guys. Her name is Nina. Nina is heckin' cute. She is heckin' cute. Y'all want to meet Nina? Y'all want to meet Nina here? Let's see. Yay. Come here, Nina. You want to say hi to everybody today? Okay, so here is Nina. Oh, her paw got caught in my hoodie. Here's Nina. She's the science doggo. So Nina the science dog. She's new in the house now. Another family couldn't take care of her anymore. So we're like, hey, yes, of course. I have Frodo and now I have Nina. She's like about the size of a shoe. So here's Kevin Smith's shoe, like for real. She's about the size of Kevin Smith's feet, just so you know. Hey guys, hi. So I got Nina the science dog. She's here to help me. She's really good at Zoom meetings. Hey Nina. I know, she's like shaking. She's so nervous. It's okay. But Nina's awesome. So she's here to help me. And like I said, she's like legit the size of Kevin Smith's foot. Look, like for real. What size shoe is he? That would be Nina. So, okay, Nina, I'm gonna put you right here. It's okay. You're doing good. She's got her own pillow now. I know. So, today, sit. There you go. She's really nervous, guys. Nina's really nervous this morning. It's just I've been working to get the show together. So this particular topic that I have today. This particular topic I have is viral testing. And my background has been in analytical chemistry. And so everything to do with testing and diagnostics and that sort of thing. I know there's a little tail wag. Look at her. She's so cute. She has like a cataract in her eye, but we're gonna see about getting that fixed. I know. Are you nervous? Are you nervous? It's okay. She's so cute. Anyway, so this particular episode, we're talking about viral testing. And viral testing is really, really huge right now because we've got the coronavirus. And so I thought I could talk about all of the different types of tests. How do we test for viruses? Which ones are the ones for when? Is there a time frame? How accurate are they? What about false positives and negatives? Yeah, she's a cutie pie. Let me say hi to everybody in the chat here. Who we got? We've got Be In Mindful 71. Yay! You are the first one here. First. And Missy Ann's here. Yay for alarms. I was on Pimp Monk and Missy Ann's show yesterday over on the Morning Wood. We had a lot of fun. So some shout out to Pimp Monk and Shabisky and Ronan and Missy Ann was there. It was a lot of fun. We had lots and lots of laughs. It was great. Hey, Mr. Shadow Kitty. Virtual hugs to you too. Mistaken Reaper 22. What's up? We got Team Zoe. That's right. Zoe's amazing. She's a wonderful producer. Hopefully when I get the Sunday show back, I'll be able to bring her on because that's the more demanding show, and that's certainly the one I need help with, and she is amazing, as Monty would say. Kristen Frost Laser Beams. Hello, how are you? Peter, gonna go for it, who recently made a tribute video for me. So I'll have to go to his channel and play it today if you guys haven't seen it. Yes, I got little... Everybody says you're doing a great job, Nina. Everybody says you're doing a great job in the chat. It's okay. You're doing good. You can tell she loves you. I know. She's so cute. She had a nightmare like the other night. And I heard her I woke up and she was sitting there. She was just, it was like her second night here. And I just started patting her on the head and I told her to be okay, give her a little hug. And then she went right back to sleep. It was cute. Hey, yellow desert rat, how are you doing today? Oh, yay. I'm so glad you guys had fun yesterday. I had a blast being on Pimp Show. It's great. So, um, so today I also have to give a shout out to Hannibal and Monty. I'm going to be on their show at the end of April. We're going to talk about the Tiger King. Hannibal and I have watched that show and we had lots of dialogue with all of that. So I have lots of comments about that show. So I'm going to save it for their channel. So if you want to hear what I have to say about the Tiger King, I'll spare you. Everybody's horrible and nobody deserves to have tigers and they should all be in jail. So you go. All right, awesome. I'm doing all right. We're doing okay. I've got my co-host Nina, the science doggo. She's here to help us talk about viruses and viral testing. So let me pull up. I don't think people realize how much time goes into this type of show. If you're a creator, if you're like a content creator, then you totally get it. But for this type of show, I do have quite a bit of knowledge associated with this, with diagnostics, as far as analytical chemistry, testing, that sort of thing. My background, I've had some research in detecting exosomes from breast cancer cells in the blood through conjugated nanoparticles and detecting them through fluorescence and Raman spectroscopy. So I have some knowledge of this, but it's still, even with my knowledge of this, it took me about 2 hours just to get everything together. So that's kind of, if it's something that I'm not as familiar with, usually 3 to 4 hours. So if you're a content creator, you completely understand how much time goes into a thing like that. Now add, somebody, even with myself, who has knowledge in certain things, it still takes a bit longer. Oh, and don't forget, I am giving away a t-shirt. I'm giving away 3 t-shirts to my top 3 viewers through Streamlabs points. We've got that. Don't forget, I still have them. I've not forgotten about you. I hadn't streamed as much last month, so I want to make certain I stream enough to where it's like kind of like that. So let's do that at the end of the month, and I'll announce it on my channel, and I'll mention it over on Hannibal and Monty's at the end of April. So top 3 viewers, there's still time to where you can get it just by watching and getting points. Hello, Dinah Cat Loves Me. How are you doing? So today we are going to talk about viral testing. Gosh, it looks like I have cut this off a bit. Let me see if I can fix that. Alt. There we go. Looks like I cut off some of my own stuff there. Now we're there. Okay. So oops, sorry. That's my script. You guys don't want to see my script. It's not a Scientist Mel show unless I have some kind of silly technical difficulties. It's kind of like when they laugh on Saturday Night Live. Those are the best shows. Okay, Streamlabs working. It should be working, is it not? It should be working. Oh, gosh. Okay. That's weird. It should be working. I don't know why it's not working. Things are, YouTube is being strange lately. That's okay. I'll go back through and assign points for people who are watching today. So I can manually assign points. And I can look and see in the chat and see who's here. And I'll give y'all points today. How about that? Extra points for coming to learn about viral testing. Okay. COVID-19. That's a thing. And it's a massive pandemic now where the lives lost are on an exponential level. U.S. is #1 now. And will probably surpass Italy's number of deaths by the end of the day. And we'll have the most deaths in the world. Now, keep in mind, with these particular viruses, population density is how this sort of thing passes. from 1 to 1. So however many people are around in an area, that's going to kind of dictate how quickly and how much a particular virus will be within a population. Now, testing being delayed caused a rise in infections as isolation and quarantine were put into place far too late. Now we're testing more and learning more about COVID-19 through viral testing, but many questions as to how these work. Miss Ann, test donations. Thank you. Test donated $5. Thank you. I don't that was awesome. Thanks for that. All right. So many people have questions as to how these tests work and why there are false negative and positives. Let's learn about the different types of tests and where science is going with COVID-19 testing. Okay. So here we go. COVID-19 news. Actually, I needed to probably change that to viral testing. Typos. I got my own typos there. All right. So, thank you, Miss Ann, for making certain it works for me. I appreciate that. All right. So, types of tests. We're going to talk about types of tests, how they work, the tests that we use for COVID-19, and possibly some new research coming out of that. So keep in mind, COVID-19 is an emerging thing and information is changing rapidly. But that's the thing about science. As soon as we get more information, we change the direction of the research and we also change perspectives. Beforehand, people were like, oh, this is just an old person disease. I can go out and do whatever I want, which was kind of selfish, if you ask me. But then once people started to develop symptoms and have pretty severe symptoms associated with infection, people in their 20s and younger, they were like, okay, wait, it's not just an old people disease. But this isn't the first time we've seen something like this. We've seen this before with HIV. HIV was originally called GRID, gay-related immune disease, because they thought only gay people had it. And we know that's not the case. That's not how viruses work. They don't just say, hey, I'm going to target this population and nobody else. There are always going to be cases associated with that where you have others in the population affected by it. But we can talk about that another time about how epidemiology works. So let's talk a bit about, hey, good morning, OGK Kush, J Kush. Let's talk a bit about what the type of tests we're going to discuss today. So the different viral tests that we're going to discuss, there's the antibody viral test, there are viral antigens, viral culture tests, and viral DNA or RNA tests. You like my hair? Thank you. All right. So we have all of these different types of tests available to us. Right now, the one that's being used is viral RNA, essentially. Diseases don't discriminate and love to take trips in humans to find more compatible hosts. Be kind to grandma, your immune-compromised friend, and your 3-month-old niece, and don't put them at risk. That's from Kristen Frost Laser Beams. Yeah, that, it's one of those things we need to not put people at risk. I mean, come on. So people are like, oh, it's just 2 to 5% of the population. Well, that's millions of people in the U.S. You have 1 million billion points. Says Kristen to Dinah Cat Loves Me. Oh my goodness. All right. So when we're talking about these particular tests, in order to understand these viral tests, we have to have a working knowledge of some cellular and molecular biology. This includes how cells communicate with each other through receptors. Receptors are particularly important in protein signatures, as some call them. Virus recognition, a little bit of genetics, and the immune response. So it's important we kind of go through some of these basics to make certain we have a working knowledge. Yeah, working on it anyway. Oh my goodness. I got a Pac-Man machine in the back. I do, OG. I do. I got a Pac-Man machine in the back. All right. So here are the different types of tests. We've got the antibody test, and that is reliant on your immune response. So if you're infected with a particular virus, your B cells will make antibodies. Now, these antibodies will attach to the virus and kind of signaling all the other immune cells to kind of go in and go. There's also viral antigens. These are the little receptors, these proteins on the virus. So if you're familiar with seeing that image of COVID-19, all those little things that look like knobs on it, these are viral receptors. And so the antibodies would attach to those, and therefore they'd kind of neutralize it. So for the test, we check to see with viral antigens to see if, they attach. If you got the viral antigens, on the virus, they will attach the antibodies, and there you go. We can also do viral culture. I can't speak today. Viral culture. This infects the cells. And so with viral culture, this is how, essentially the virus attaches to the cells through those little viral antigens. And they attach to the cells, and they're able to infect the cells and enter into them through that cellular type of communication. And then we also have genetics. Viruses, you have RNA viruses and you have DNA viruses. So inside of that particular capsule that they have, and with the COVID-19, it's a lipid-based one, so it's oil-based. And inside of that little oil-based type of package, that lipid-based package, you've got all of this genetic material. It's an RNA virus. So it injects that RNA into the cell, and then the cell starts making all those viral proteins, and viral proteins get put together to form viruses, and you have a gross birth of all of these COVID-19 babies going around just to infect more of your cells. And some viruses, they have the cell make the parts, release them into outside of the cell, and then the virus will go and put together copies of itself manually, kind of like that. And so you've got different ways that goes. So all of these different tests look for different things. Let me check up on the chat. Let's see. Hey, Tamara, purple sweet grass. Is it McGugan or McGuigan? McGuigan? Golden Coin 100, was the COVID virus even isolated? Yes, it was isolated. They're working on, now since it was isolated, now they can work on antibody tests. Where I can find the process taken to prove the accuracy of the PCR test being used today for COVID. I'm going to talk a bit about that. I've consulted with a couple people who work in the field, and they gave me a little bit of a rundown. Concordance was one of them on Twitter. Gave me a bit of rundown on how effective PCR is over a period of time. Yeah, I'm just really getting into it, so we're good. Alrighty. Where is the gold standard this test was created around? It depends on which test you're talking about. I got that a bit late, so be a teeny bit more specific with that, and I can get you that information. So let's talk a bit about how antibodies are made. Now, our natural immune system will make antibodies that are generated from our B cells. And so let's have a look at this here. Okay, so here, for instance, if you were vaccinated, which I hope you were, if you're vaccinated for the mumps or measles virus, and if you're not certain how vaccines work, I have a show for that. Check out on my channel, youtube.com/ScientistMel. You're here now. And if you're not, and if you just showed up, don't worry. Check out that episode on vaccines. All right. So, yes, I know I'm touching my face, but I've washed my hands a lot and I don't leave my house. So, I also disinfect everything that comes into this house. So, we all right. Hey, Nina. This is Nina, the science doggo. She's already tired. She's already tired. So, if you have been vaccinated, your body has made antibodies associated with the non-infectious version of the virus. And so what happens is if you were exposed to the mumps virus or the measles virus, these antibodies are already floating around in your blood, and they attach to those viruses, neutralizing it, preventing it from entering into your cells, and then the virus is killed from the other immune cells. They go around and they're like, hey, this one has been targeted. This one has been targeted, you know, and then they're able to go through and knock them out. Okay, let me check out. I was working in this hour to bring a vaccine. Don't make any quick moves. There's a wolf-like creature stalking you, says Michael. Oh my goodness. McGuigan. Okay, McGuigan is how we say it tomorrow. Okay, awesome. Sweet. Okay, so this is kind of the rundown on how our bodies make antibodies. Very, 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 simplified explanation of that. With now, how in the world do we test for antibodies? So let's say I've been exposed to COVID-19. Right now, they're developing an antibody test. An antibody test would be a bit more sensitive. So with antibody tests, this particular test, what happens when you get tested to see if you've been exposed to a particular virus, it looks for a specific viral infection. So usually what happens is it's done with a blood sample because your antibodies are kind of floating through your blood. So if the antibody is found, then it can show that a person was infected recently or in the past. Now, we usually use a test called ELISA, enzyme-linked immunosorbent assay. And this pretty much confirms if there's exposure to the virus, but it doesn't necessarily mean that the person is currently infectious. So that's one of those things. You can have antibodies against a particular disease and not be infectious or spreading it actively. So antibody tests work great to let us know that, yes, you've been exposed to this disease. Yes, you do have immunity to it because you have the particular antibodies, but it doesn't tell us whether or not you're infectious. But once we learn more about diseases, particularly about COVID-19, it will let us know a bit more about when the viral shedding happens. Still, you have to form the habit of not touching your face, so don't do that. Yes, Samika Nuts. Oh gosh, I've seen a lot of conflicting things about exosomes in COVID SARS too. So that's the COVID-19. I've been watching you from Omni Eris. Oh, thank you, Tamara. Yeah, I adore Omni. He's great. He and Doc are awesome. Doc is a very good friend of mine. So they're awesome people. And they're very good guys. They're part of the good guys, All right, so how does ELISA work? So now that we have isolated COVID-19, we're developing an antibody test. And so what we do is we take the antigens off of the virus and we bind it to a particular surface. And once we bind it on that surface, we can run our clarified blood sample where, we have to spin it down a little bit. Hey, Jen's little corner. So we have to spin down that and isolate the antibody. So we can just essentially separate the red blood cells from the antibodies with a centrifuge. And then once we collect them, we can take those antibodies, and so we have an enzyme attached to it. So it's an antibody enzyme conjugate. So they come together, and so they pass through, and you run it out, in an ELISA, you can like inject it in there. And then if you have antibodies for the particular virus in your blood, they'll stick to those antigens. Because remember, that's kind of how our immune response works. Those antibodies stick to the antigens because they're the right shape for them, so they have the right interactions. And so if your antibodies are in the blood, they'll stick to those antigens. Then we can put like a color on it. So we let it sit there for a minute, and you're like, okay, should have, depending on, usually, it really depends on, it really just depends. It could take 30 minutes to a couple hours, to stick it on there. Just depends. And then you add a dye. And so the dye is a substrate and enzyme interaction. That dye will attach to that enzyme, that particular enzyme that's stuck on the antibody, and it lights it up. And then we can use like this little microplate reader, run it through, and it looks for that color. So if that color is there, because it'll only interact with that enzyme, then that means you have antibodies for the particular virus. That's how that works. Let me catch up on the chat. Hello. Jen's Little Corner's here. How you doing? My partner wants to know, is it airborne? It's not airborne, but it can live in aerosols. So aerosols, we think of it as like a spray, like an aerosol can that you can spray stuff with. Well, people can sneeze and cough and generate aerosols, just like a hairspray. And you're just like, that's kind of really a gross concept. But aerosol droplets, we've found that with COVID-19, it can live in aerosol droplets, which usually are about 5 microns. So how big is that? It's about 5 times the size of a cell. So droplets that are about 5 times the size of a cell can hold COVID-19. This is why that N95 mask is particularly important, because N95 prevents certain tiny, tiny, tiny particles from passing through. Now, cloth masks are great, but they're not meant for extended, super strong type of protection, and you need to wash them. If you're just popping out to the shop, cloth mask, that's great. It'll catch some of those liquid drops, but you still need to take it home and wash it in some hot soapy water. You can't just use the same one over and over again. It's kind of the idea with like people who wear gloves and touch everything. They touch this, then they touch their keys, then they touch this. They're like, I'm wearing gloves, I'm safe. Well, if you touch a surface covered in COVID-19, then you go and you touch your dog or your cup or your keys or something like that, you're just passing it around. If you're touching like a basket at a store, and you've touched all of the products that go into the basket with your gloves, you've touched the basket, you've touched your products, you've touched your wallet, you've touched your keys, you're gonna have to wash everything now. So it's probably just easier just to wash your hands. If you're going to wear gloves, great. Don't wear them everywhere. Don't wear them all the time. Change them frequently. You go, you touch a thing. Oh, I touched a thing. Time to throw these away. Put on another pair. This is what we call reducing contamination or cross-contaminating. So there you go. Let's see. Okay. Catching up on the chat. Just for the record, I'm Michelle from Atheist and Junk Shop Library in Nervardia, and Jen's Little Corner, and a bit of tea in Danish, and Critical Cripple. Golden Coin asks, why is it so difficult for them to get antibody tests out? That's a good question. First of all, they had to isolate the virus. That was the first thing to do. Now they're able to infect mice. Now, Okay. I'm trying. Let me think about this. Which way do I want to explain this? All right. So now that they've isolated COVID 19, they're able to get the antigens and bind to the surface. Now they've got to figure out which antibodies are going to give the strongest detection response. Usually it's usually it depends on the different shapes of the proteins on the outside of the virus, because they could have different combination of shapes, and your antibodies will develop to fit onto those different shapes. And so what they can do now what they can do now is they've got to test and see how effective their antibody test is, and that takes a little bit of time. So if they're able to infect mice or other types of smaller animals, and get a solid bunch of antibodies, and then they're able to test the efficacy of it, and how well it works, after a period of time. It takes a little while to show the effectiveness of a particular test. It takes a little while. If they're able to draw antibodies from other patients, they're using that as a form of immunotherapy for patients who have been exposed but recovered, transplanting those antibodies into somebody else. But that's one of the immunotherapies. It's an old therapy back when they had the Spanish flu going on. Just antibody donation and transplanting antibodies from one person to another. So that's one thing. It takes a while to definitively show, and you have to have a control, and you have to have an infected thing. So they have to show that the test is more effective than the control by a certain margin. But once they get it going, the antibody test will be good at helping us track who actually has been infected. You know, PCR tests are limited within a certain window. You only have a certain period of time to detect whether or not a particular virus has infected your cells. They stop being effective after seven days. They're not as effective, and then you tend to have a higher false negative rate. But antibodies, if you have the presence of antibodies in your body, then that says, yes, you have been infected. And if you are asymptomatic, fantastic. Then we can start testing people and see, okay, the ones who are asymptomatic, do you have allergies? Give us information about your day-to-day. What's your health history? You can do lots of information, even, with that type of thing. It's not as strong with PCR testing, although that does let us know definitively which strain and which virus you have, because it's genetic testing. All right. Okay, so your partner, Tamara asks, he does most of the running around, and his parents are in their 90s. Should he be wearing a cloth mask shopping, or should I get him another kind of mask? You can wear a cloth mask and that be okay, just if it's the kind that's reusable. Be certain. Okay, so I'll tell you my protocol. Because one of the warehouses out that I buy my groceries from, and they stock about 100 different stores in my area, or 100 different stores in my state. Let me say that. They have workers that have tested positive for COVID-19. And so we're relying on the grocery store. So what I do, if I have to leave my house, I have clothes that I wear that are my outside clothes. And, shoes, you can spray the bottom of your shoes with a disinfectant spray if you want. But I have clothes that I wear outside that I put in the washer, and I wash when I get home, and I take a shower when I get home. If I have groceries or stuff like that, if they're in cardboard boxes, I might spray down the cardboard box briefly, but honestly, it's probably not going to do much because it's cardboard. But I take out and dump out the stuff on the inside. Um, I disinfect all my cans and, my products, stuff I get from Amazon. I do the same thing because, people are testing positive for it. So I have clothes that I wear in my house, and I have clothes exclusively that I wear outside. And I throw those clothes, if I go outside, go to the store or whatever, leave my house, I throw those in the washer when I get back home and I take a shower. I don't particularly wear a mask, but if I did, I had a cloth one, yeah, I put a cloth one on. That's mostly to protect other people from anything you might carry, because we have asymptomatic people. It may not necessarily 100% protect you from getting the virus, because we see that with healthcare professionals who are actively around COVID-19 positive individuals, but they're also using those cloth masks. So the mask is mostly to prevent you from spreading it, but wash it. It might have some minimal protection against aerosols, but you just need to wash it in hot water when you come home. Throw your clothes in the washer, wash it in hot water and soap every time. That's kind of what I do. Alrighty. Hey, bigzebra.com. Okay, Thomas asks, how many particles does it take to catch COVID-19? For example, if I were to be introduced to the smallest detectable amount, would my immune system fight it off successfully? Thanks, Mel. We don't know, Thomas. I don't think we know 100% just how much, because we have asymptomatic people. Maybe they are exposed to the smallest undetectable amount. You know what I mean? Until we do more broad-scale testing, we're not going to know 100% how that works. And it could possibly be different in other individuals. We know that people who have who smoke, their lung cells are more susceptible to COVID-19 uptake. So that's what we're learning. So with COVID-19, if you're a smoker, you're at an increased risk of infection, because those cells, it's easier for COVID-19 to infect them. So there's a lot of different factors that could play a role in that. I'm not certain about non-smokers, because as before, we learned that those that have a suppressed immune system are immunocompromised in some form. The elderly, individuals with autoimmune disease, people with HIV, or suppressed cancer patients on chemotherapy, they would be at risk. So initially we thought, okay, those are the ones that are developing the severe symptoms that are lethal. Everybody else is fine, but that's not the case. Completely healthy individuals are dying from this, otherwise healthy, with no underlying comorbidities or other diseases. So it's hard to know. It's hard to know. I'm trying, so it's just be in mindful says, I personally see some crazy crap with people who are wearing masks and gloves. I mean, it's kind of like I posted a meme on Twitter. I'd seen it on Facebook. It says people who wear gloves everywhere are like a person that uses a condom with different people. You're just spreading it around. So it's like, for some people who don't know how to use gloves, they can make everything worse. So it's just, if you don't know how to properly use gloves, just use hand sanitizer all the time while you're out. Just do that, or wash your hands all the time, because you can make things a lot worse and infect your own house, because we know it can live on surfaces for like a couple weeks. So similar to the flu. Alrighty, let's see. Hello, Benjamin. Goodness. All right. You're welcome, Tamara. I hope that helps. Cashiers wear rubber gloves. It only protects themselves when they tender cash and give you change. Yes, money. Pro tip, guys. Don't pay for your groceries with money. And also, you can take a Q-tip for like your little, when you do the card, slide it in there and use a Q-tip and type in your numbers. You can do that and then throw your Q-tip away. Don't use the same one. Just throw it away. Q-tips are pretty freaking cheap. pretty much biodegradable if you get the cardboard ones in the middle, you know what I mean? Just throw it away. So that'll help. Um, yeah, so money can, yeah, don't use money. If you can avoid using money, just don't do it. Oh my God. My daughter has chronic asthma and has extreme social anxiety. This whole thing has scared the bejeebus out of her. I am so sorry, Jen. I hope she's okay. What is it, do you think that, is it the hydroxy, is it hydroxy? I think it's hydroxychloroquine, maybe doing on a cellular level to help those who are most sick of this virus. We don't have enough information on that. There are people, that study that was passed around is a train wreck. It's a very small sample size. The methodology is questionable. The conflicts of interest are tremendous. It's scary. And from my understanding, the drugs that the administration wants to mix together can cause heart problems. What I find particularly interesting and troubling on not only a scientific ethical level, but also a humanity level is that they're going to start giving this to prisoners. So we might be seeing another gross act of human cruelty on subjecting people in the prison system to a highly questionable, not tested enough drug that can cause heart problems and can be lethal, and often is. So we got to be careful with that. It's okay. Don't worry about it. Golden coin. Let's see. Yeah, there isn't a lot of solid evidence on, the efficacy of, I think, is it hydroxychloroquine? I can't remember right off the top of my head. See you later, Peter. Thanks again for your tribute video. Big Zebra says, do you ever play the Pac-Man behind you? Why not play Ms. Pac-Man? That's more thematically and politically correct these days. Oh my gosh. It's I do. I played it a little bit. That was a gift from my brother. There's actually Galaxa and Galaga on there too. But it's kind of nice to have it playing in my background. It's pretty neat. Oh goodness. Let's see. Why isn't it Mr. Pac-Man? Oh gosh. So I'm trying to catch everything else. You're welcome, Thomas. I'm glad I could help. After you quit smoking, do your lungs ever fully recover? That's probably not. I'm going to say it's more often no than yes. You can have a reoccurrence, depending on if you have cells that, particularly with lung cancer, you can have cells as you age and you get older. Your immune system tends to shorten. You don't make so many antibodies as you used to. So your immune system is the thing that takes out cancer cells. So as you become more immunocompromised as you age, those cells that were affected by, if they became cancerous in the past and your immune system was killing those particular bad lung cells that were pre-cancerous, you might still have a few that are hanging out. So I've seen that happen with some people who used to smoke and then they started smoking. They stopped smoking and then they still managed to get like oral cancer or throat cancer or lung cancer and things like that. So it's hard to say. You certainly reduce your risk of that, but it's really important to kind of have a working understanding. So if you think that might be a possibility for you, make certain you get a checkup every year. And, they can do a chest x-ray and things like that to kind of see where you're at. About what to do with fresh produce. Honestly, I just use soap and water. So people are like, you put soap on your vegetables? Why not? And I know it's porous. I know it's porous, but getting a little bit of soap isn't gonna hurt me. It's not. If I digest a little bit of soap, a teeny bit of soap, it's not like I'm sucking up bleach or Lysol. You know what I mean? But I just thoroughly wash my vegetables with a little bit of soap and just scrub them all down. Vinegar's not going to kill the virus. Soap or some kind of emulsifier that eats up oils, that's a good go-to. So that's just kind of what I do. Yeah. What's my thoughts on UVC lights for killing viruses? Ultraviolet light, yeah, that's pretty good. It depends on, I think it just really kind of depends on the virus. But if a virus has to live in fluids, all you really need is a disinfectant, honestly. If it's like an airborne thing, usually ultraviolet lights tend to work pretty well with those. I just have my son thoroughly wet out the money with 70% alcohol and let it dry for 2 hours and into a fresh envelope. We use that to pay for deliveries. Yes, you can. That's absolutely fine. That's a good idea. Alrighty. The Pac-Man game that Paul on Periscope says, the Pac-Man game is how I feel when I try to take a walk while social distancing. Scientist Mel, your background is an excellent example of the concept of visual interest. Yes. What is that back there? And I wonder sometimes if it's counterproductive to what I'm doing because people want to know what's going on behind me. All right. Now, we've talked about the antibody test. Let's talk about the other tests really quickly. What about the antigen test? Okay. So, Galaxian and Gyrus are my two favorites from the era from Jen. Oh, you saw my baby? Yeah, here's little. This is little Nina. She says hi. Wash your hands. Yeah, wash your hands. So I'm assuming if you cook the food to a certain temperature, then it will kill it. Yes. If you cook it to a certain temperature, yes, it will kill the virus. You'll be fine there. That's how it. Yeah, you can do that. Alrighty. So let's have a look. Let's have a look at this. Let's talk about the antigen tests. Alrighty. Yeah, Nina is my co-host now. So, Nina the science doggo. She's pretty good. She's a good. Here, and there goes Wonder Woman. See, that's the downside to having things. I'll get Wonder Woman later. She's strong. Nina's going to investigate and make certain she's okay. It's all right. Sit. Okay. You want to come over here? All right. Nina wants to sit. All right. So, now with an antigen test, what you can do is you can take tissue from essentially you can take tissue from your patient. So it depends on the type of tissue. So if it's like COVID-19, we're doing like PCR with COVID-19. So you can take a particular tissue. So depending on the area that's infected, whether it's stool samples, you can use stool samples for that. You can use nasal cells, throat swabs, that sort of thing, or any kind of if it's an epithelial based type of virus, meaning that any kind of tissue that lines something, like epithelial being skin-like tissue. What happens is you have antigens that are expressed on the outside of infected cells. And so once you get antibodies, then you can take that particular tissue and you can run these antibodies, and usually you can buy them. So I used to work in testing and diagnostics, and we could generate antibodies from different animal sources. And so what they would do is they would add this type of fluorescence type of dye onto these antibodies. And so once these antibodies attach to those antigens, the presence there, you're like, okay, then you could definitively show that this person has that particular virus. And so those tagged antibodies kind of show up under a microscope, or even on a certain wavelength. You can kind of blast it with light and then it'll light up. And so that's what that dye does. It lights up to make it easier to spot. Now with indirect immunofluorescence, you still have the same antibodies. You have antibodies that will attach, in this case, you have laboratory cells that are infected with the virus. So you actually infect the cells. Then you take the patient's sample and see if they have, antibodies. And then those antibodies could attach. It's kind of like ELISA, could attach to those antigens on the cells that you infect with the virus. And then they can tag those patients' antibodies with other antibodies with that fluorescence on there. And then that lights it up too. So this type of detection test, it's pretty useful. I mean, in the where it attaches to the actual viral antigen, but we have to already have had antibodies developed, whether through an animal source, which is usually the case because we can make a lot of antibodies from like goats and sometimes mice, but usually it's goats. They use goats for that. Okay, let me catch up on the chat really quickly. That's the other type of test. Nina. Nina the science doggo. Nina, lady. UVC lights on viruses. Thoughts? Yeah, I mentioned that. It could for some of those be if you understand the wavelengths. So UV light can affect molecules in a certain way to where it can actually kind of bust up certain types of bonds. And so that's why, we're susceptible to UV radiation. It can actually mess up our DNA. And so if it's able to kind of mess up our DNA, well, it could conceivably, as far as I know, it can mess up viruses. Alrighty. As far as I know, it can mess up viruses. So, I mean, there's a certain amount of energy that's associated with UV radiation, and that energy is enough to bust up certain types of bonds. So it can do that. Yeah. My partner is obsessed with an air purifier, but never turns on the UV, which makes no sense to me. Turn on without the UV. Well, I mean, an air purifier can remove things like pollen and dust and things like that. So if you have a tendency towards asthma and allergies, particularly if they're allergy-based, that's a good thing to have because it can remove those particles out of the air and make it a bit easier for you to breathe, with that stuff. Okay. Let's talk about viral culture. Viral culture, not like, couture with all of the fancy face masks that come out. Did you see that they make face filters now? They make like face mask filters for different camera apps. You can get a fancy face mask and be like totally fashionable. And I'm like, what the heck? No, so this isn't viral culture like that. This is an actual test. So here we go. Okay, so this is kind of how we go through. We will take a sample of healthy tissue. So when we're doing virus testing, we're looking to find a virus that can cause an infection. So we will take a sample of bodily fluid or tissue, and it's added to certain cells to help it kind of grow a virus. And so depending on the virus, what would happen is if it was, again, like one of those epithelial types of viruses where it could mess up with it infects like skin-like tissue, like stuff that lines things. Or if it was like a lung cell type of virus, we could take, lung cells, grow them in a dish, and then we could take the blood or essentially the patient's sample, usually it's blood, spin it down and see if they have a virus that infects that tissue. And so then if it does, you can do various tests and, usually like with a microscope, you can see like infected cells and what they look like versus non-infected cells and compare, and then that can show you that you have a virus. Now, the thing is, this can take several weeks to show results. So it's not really a fast test. And so after a few weeks, hey, this person had the virus. Oh, they're dead now. That sucks. Or, oh, they've recovered. Fantastic. So, it's something that isn't always, my nose itches, isn't always something that just, Yeah, so let's see. Okay, I think I've caught up on the chat. All right. So how are you guys doing? We doing all right? Okay, now we're at the last version of this. Okay, Nina, I'm going to put you back here. I think you're doing a good job. Let's have a seat. Stay right there. We'll investigate Wonder Woman in a minute. She fell down. Okay, so the last test that we're going to talk about today, and then we'll get into COVID-19, how that test works for them, the pros and cons, and we'll talk about false negatives here in just a bit. Alrighty, so viral genetic tests. This is what happens. All right, so if it's an RNA virus, I've got hair in my mouth. Okay. If it's an RNA virus, we have to use something called reverse transcriptase that goes through and converts it to DNA. Now, we add dyes to that DNA, and then we add something called primers. So there's certain sections of that DNA that is a defining section that says it is this particular virus, because viruses have slightly different genetic makeups. And so where that difference is, that's where we put our primers. And that section is called, we amplify it. So essentially all that means is it's like making a photocopy of the DNA. We just make a whole lot of copies of it, a whole bunch of copies over and over and over and over and over and over again. And, you know, it could take a couple hours. Go through, make all those copies, and then we run it through a gel electrophoresis. And so what happens is if the gene shows up, or if the DNA shows up for those particular molecular weights, and you're like, hey, look, we got it lit up on these three patients. It's 2, 4, and 5. So the example here, let's say that this is the particular band that you would expect to find associated with this particular virus. And you're like, okay, patient 3 doesn't have it, but patients 2, 4, and 5 do, because it lit up on our gel electrophoresis. So if the sequence is present, that means they're positive. Okay. So you're like, up until now, I thought this was a fear tactic. So I'm glad I caught your show. Yay, Tamara. I'm glad you caught my show too. So this is how the genetic tests work. Now, let's talk a bit. Let me go to the next one. All right, so I found a really great graphic, and this is from compoundchem.com. And we're going to kind of break it down. So thank you to Andy Brunning from Compound. This is Compound Interest 2020. And you can follow them on Twitter, Compound Chem. They're also on Facebook. Have a look at this. Now, I know you guys can't see this, so I've broken this down, but we're going to kind of go through how do the tests for coronaviruses work. So right here, this is how the current test works. And so what they're doing is an RNA test. It's the genetic test that I've mentioned. So a swab, you take a swab and it's in the nose or in the back of the throat. It's not particularly pleasant, but it works. You know, you get a swab and then it's sent to the lab to test. And so then the RNA of the virus is, you know, extracted, it's purified, and then they convert it to DNA through that reverse transcriptase. And then they add the primers with the dyes. Then it makes all of these copies of the DNA. And so it gives off more light. So I showed you an example of a gel back a little bit ago. So it lights up. So if the DNA is there, if the DNA is there lit up, then that means a positive presence of the viral RNA. You know, that means the virus RNA is there. So positive and negative tests. So that fluorescence, it increases as more copies of that DNA is added. So it gets brighter and brighter and brighter and brighter and brighter. So it crosses a certain threshold. So that threshold is what we call a limit of detection. And so we have to amplify the DNA in order to get it to show up. Otherwise, it would just be such a small amount, it wouldn't show up. So the idea is, if the DNA is there, then the primers will attach to it with the dyes, and then boom, big old bright light showing up on your gel, right? So if it's a big bright light, it's positive. If there's nothing there, it's negative. Now, issues with this type of testing. Right now it's in high demand, and so it's really hard to get all the reagents that you need. it can take a few hours to run one of these tests. And depending on, you know, the size of your PCR machine, this can also limit how many tests can be done at a time. Now, you can have false positives and false negatives. More than likely, you're going to have a false negative because the sample can degrade. There's a risk of contamination because you have to handle it so many different ways. The more times you got to handle something, the increased risk you have of contaminating the sample. There's also a time frame. And so time, if it's greater than 7 days, you're likely is that of onset of symptoms and sometimes even exposure, the likelihood that you'll have a false negative increases. All right. So future tests, they're working on antibody style tests. The current tests are good about letting us know that there's an infection, but they don't tell us if somebody recovered. You know, they don't tell us that. But the antibody tests would. And so that would definitively show that, yes, a person has been infected, but, you know, and, you know, whether or not they're still recovered or not. So these are the different ways that this kind of works. Let's see. What is the false positive rate right now of the PCR test? I'm about to get into that. So Concordance on Twitter had a little private, you know, chat with them to ask them, you know, hey, you know, I noticed you were tweeting a lot about false negatives and that sort of thing. The largest thing it has to do with is the window. All right. Let's see. The largest thing it has to do with is a window. All right. I was just checking up on the chat really quickly. All right. So false results. What's that about? All right. There's different things that can happen with false results. If you already have a strong immune response, a strong immune response can, you know, already kind of wipe out the stuff, and then so you would lack a certain amount of RNA there. So if your body has effectively fought the infection, now we think that in one working hypothesis for this is that if your immune system is fighting off the virus, you're reducing the viral load, which means there's less RNA around in order to effectively test for. Contamination is a problem. Your sample, if it sits around too long, RNA falls apart pretty rapidly. And so it doesn't fall apart as quickly. I mean, it falls apart more quickly than DNA. And so if it's sitting around too long, if it was contaminated, if it got mixed up with something else, contamination could be a potential issue because the process of PCR requires so many steps. It requires a very high level of skill for individuals to not cross-contaminate and also to not leave a sample sitting around very long. You have to convert it to DNA pretty quickly, and then the amplification process. And in PCR, it requires it's a specific temperature. It's polymerase chain reaction. That's what PCR stands for. There's a particular enzyme that's found in certain bacteria that live in extremely hot environments. And they found that they're able to replicate things pretty fast. And it's at a heat that doesn't degrade the DNA. And so it's able to go through and make all of these copies pretty rapidly. And so also inappropriate test windows. You have a harder time, particularly with my nose itches. Now that I can't touch my face, my nose itches all the time. It's like a psychological thing. But now there's like inappropriate test windows. If you test later than 7 days, you know, you're likely your chance of getting a false negative increases because of the fact that the RNA just isn't there. So here's an example. So Concordance sent this to me. He sent this to me and he's on Twitter. Check him out. So nasal and throat tests. Once you get about to, you see right around, If you're at 30 days, you can forget it. So it's a month later. So if you have people, particularly that are asymptomatic, they're not at the high level of we should be testing people who are asymptomatic. How do you know? How do you know when you are exposed to the virus? Exposure and onset of symptoms, infect a patient depending on when they were infected. Some people tend to have symptoms show up at 3 days. Some have 2 weeks. So the onset of symptoms is the clue. Once you get symptoms, you go and you get your PCR test, because the longer you wait, the less likely you'll have a good, the more likely you'll have a false negative. That's essentially what that would be the way with that. Alrighty. Yep. So, Mel, do you subscribe to Netflix? If so, could you find some hidden gems for us? I do subscribe to Netflix. I've almost done with Netflix, guys. I saw there was a meme going on around. I have finished Netflix. I do like Netflix. It's a lot of fun. Let's see. It's I see you're talking about the scare tactics. So tomorrow you must be in the UK, your prime minister and all that, treating this as how long it takes for real information. I'm trying to, I try to, and sometimes I'm not correct. Sometimes I mess up, and people will tell me, oh no, there's this new thing that came out, and it's not a mess up, that's intentional. And so, like yesterday, I was on the Morning Wood, and somebody mentioned, well, we're seeing people who overcame COVID are now testing positive again. But there's a lot of reasons that could be happening. And I wasn't certain. I hadn't heard about that yet. I'm like, well, we don't know. And somebody in the chat was like, hey, here it is. And I'm like, thank you for this information, because it's moving pretty rapidly. And so as soon as we get new information, it shifts the course of where research is going. So we're finding in some cases that people are testing positive again. So we don't know, is it a reinfection or is it the same infection that has come back again? Because that can happen. We see that with different types of viruses. HPV is an example. You can have you can test negative for HPV for a very long time and have it pop up. And there's lots of different reasons. Is it the same infection or is it a new one? Because HPV could be latent like that. So we're still learning a lot about COVID-19. Now, the flu mutates. And so anytime you get the flu, it's a brand new infection. That's a completely different strain. We don't know if COVID-19 is like that yet. We don't know, is it like the flu where we have to, monitor it and develop vaccines? And people are like, well, people die from the flu every year. Yes, but it's spread out over the course of a year. And we do have vaccines that help lower that number. We don't have vaccines for COVID-19. And with the current rate of infection, we're seeing a high death rate in a short period of time. Now, the death rate can drop, but if that would be with vaccines, with social distancing, things like that. We want to spread. Everybody's probably going to get this virus, guys. I'm not going to lie to you. And about 95% of people, if they develop symptoms, they'll be mild, and it's going to express differently in people. And some people may not even have symptoms at all. 95% of us will be fine, but that 5% is millions of people. And we need to spread this out over a year. If we're all gonna get it, we need to spread it out over a year so we don't overwhelm our hospitals, because that's what the issue is. People, yes, are still going to die from this, and that's a horribly sad, awful reality if we don't get a vaccine, and that we're still 18 months from that, probably best case scenario. But the testing for antibodies, then we'll be able to start testing for people and see, okay, were there people who got it and were asymptomatic? We need to know what's going on with them. let's check their genetics. Do they have a gene associated with it? What's their immune response like? Do they have allergies? Do they take any other types of medications? We can start addressing a lot of those, but we can't rely on a PCR test for people who are asymptomatic because we don't know when that person was infected. But if somebody was exposed to an infected person, we test them right away. And then if they don't develop symptoms, then it becomes useful. So it's kind of like one of those things, but it's okay. I do like Netflix. You need some new ideas of what to watch on Netflix. I'll think about some of those. It's Justin Trudeau. I'm sorry. Oh, goodness. All right. Justin has an awesome beard game. He does. All right. So, just to kind of talk a bit about the false negatives. Let's kind of go back through in case you missed it. A strong immune response can mean less RNA for appropriate testing. Now, again, concordance, thanks so much. Also, nods to Doc Savage. He is immunology and epidemiology is his bread and butter. So I have to give nods to him for keeping me informed with information. So thank you, Doc Savage, for your contributions to my work in the intellectual space. I appreciate you. And to Concordance. Thank you so much. All right. So there are 2 papers that were floating around out of Wuhan early on that were stating that there is a 40% false negative rate with COVID-19. Now, the methodology with those particular papers, are highly questionable, particularly if they're the ones that are in the preprint that haven't been peer-reviewed, and there's a lot of those floating around right now, guys. So please be cautious. Those are really good for researchers to have a look at to kind of see where they're going with, and use for data-wise. But if it's not peer-reviewed, there's still potential issues, Okay, so for all intents and purposes, 40% of a false negative rate is the absolute worst case scenario. And even then, it's, not likely. The current test is really only appropriate for under the first 7 days when symptoms present. And since it takes a while to test to COVID-19 through PCR, not everyone is being tested. Now, with many who seem to carry the virus but are asymptomatic, it makes them not solid candidates for testing, given the likelihood they may not test positive at all, as they may have been carrying it for an extended period of time. Now, the antibody test, that would be a better test to determine if someone was exposed to it. It doesn't, however, tell us if the virus is infectious or spreading at the time, like how it would be with shedding. It doesn't tell us about that. So there you go. Yeah, Doc is awesome. He's a dear friend of mine. It's absolutely lovely. Got to spend some time with him in October. It was cool. He's a good friend. He's a very good friend, and he's a super smart man, like brilliant level. So and Doc is great. He's a super sweet guy. I have nothing but wonderful things to say about him. All right. So that's kind of where we're at with testing. So I have been looking at, and if you're curious, go check out compoundchem.com. They have a lot of visual resources. Here's an example. That's the one that I found, Compound Chem. I've checked out, I've got a lot of different sources. Let's see, I didn't read that one. NIH is really great, the immune defense, and it talks about viral activation. This is a moderate level of read, and it does have very sciencey sounding terms. If you have a working knowledge of biology, with some of the terminology, you can have a go with this. This talks a bit about immunology in that regard, and the immune defenses associated with that. Here is 1 in 4 coronavirus carriers could be asymptomatic. And so this is from Science Alert. I haven't gone through this just yet, but But, 1 in 4 is pretty high. 1 in 4 is pretty high. But Science Alert tends to be a reputable source. So on Tuesday, and this came out last week. So carriers could be asymptomatic. Now, asymptomatic, that's a pretty loose term. I have allergies year round. Okay, I have allergies year round. For all I know, I could be carrying this virus, and it might present as an allergic type of response with a runny nose. I've had a dry throat for probably 2 weeks. Is it pollen? Is it COVID-19? I have no idea. And I'm not going to waste resources going to go and say, I need to be tested, because that test needs to be for somebody else who's experiencing symptoms. That's where I stand on that. But there's significant transmission by people not showing symptoms. So that face mask that you're wearing, it's to protect other people, really. Because if you're not exhibiting symptoms, you could still be carrying that virus. So you can, and that's the problem. It's going to be harder to contain, because people who think, oh, I'm well, I'm not sick. Yeah, no, well, I can go out and party. No, you can't. You could still carry the virus. So this gets into a bit of more of the studies. The Diamond Princess cruise ship, they did a study on them. And Redfield, I think he was from the CDC back here. Robert Redfield, yep, from the CDC, was talking about how people are significantly shedding the virus with about 48 hours before symptoms appear. So you could be asymptomatic and actively spreading this virus and not have symptoms for like another 2 days. So this is why it's important that we kind of stay home if we can. Stay home. Now this source, I'm skeptical of this. Stat did basically posted. They did an article based on one of those studies that haven't undergone peer review yet. This is where we run into the problem, and where I particularly, oh gosh, I told you I'm gonna wash my hands in a bit. I know I shouldn't touch my face, but you know what? I'm just anyway. The study that they discuss here has not undergone peer review yet, and I had a look at the study. Extremely small sample size. Most are likely not infectious after recovery begins. That's great, but I went and had a look at this study. So this is not peer reviewed yet. This is a preprint. Okay. Again, this is useful. This is useful for other scientists to have a look and say, okay, what did they find out? Maybe I can apply that to what I'm doing. But it's not so great for anybody who doesn't understand how to read one of these. If you look at the size, and they go through active replication, neutralization, characters, and all that, blah, 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 it goes through. I'm trying to find, they had a small sample size. I was trying to see, where was it? I was trying to see average viral load, 5 days. One of these studies, had a small sample size. Was this the other one? No, it was in here. I thought I saw it. I thought it was in this one. I have been reading so many papers, guys, like for real. However, background, current case definitions, lower methods. We study viral loads of cell culture, respiratory tract, clinical sensitivity. Was it this one? Let me see. Where was it? Is it the same one? I'm so confused now. No, it's the same one. See, one of these had only like five patients studied. Only 5 patients were studied. I don't know if it's this one. Shedding of viral RNA from sputum outlasted end of symptoms. I'm trying to find. There was one of the studies. Okay. The present study shows that the COVID-19 can often present as a common cold-like illness, can actively replicate in the upper respiratory tract, and is shed for a prolonged time after symptoms end, including stool. I'm trying to see where their sample size was. Was it in here? Metrics. I want to know. Let's preview this. Where's, okay, so that's it. Virological. That's this one. Okay, so this is the actual paper. I'm trying to find their sample size. See, that's a problem. I can't seem to find their sample size. All patients were initially diagnosed. Well, how many patients did you have? This came out of Germany. I want to know how many patients they had. See, that's the problem that I have. The HERE study patients were enrolled because they acquired their infections. What's your sample size? See, that's the problem I have. I'm like, I want to know what your sample size is. How many patients did you study? Was it 5? Was it 1,000? Was it 100? And I'm not able to find confirming each other's results. The HERE study, okay, how many patients? Okay, so one same patient. Okay, here, in throat samples, sequenced distinct virus populations were consistently detected in throat and lung samples of one patient. See, that's the problem I have. How many patients did you study? And so I'm like, so they found it in one patient, but that doesn't mean it's a trend. So, I mean, that's the problem I have. I've got to go through. I want to know how many patients they studied, and I'm trying to find that, and I'm having a hard time finding that. I described these observations in terms of HERE studied patients. How many? How many patients, Germany? How many patients did you study? Where are your charts? Where are your charts? I don't even see your charts. See? This is what I have a problem with, where media runs with these types of 2 patients. The 2 patients, they have one here that says the 2 patients who showed some signs of pneumonia. 2 patients. Sample size. So far, I've found a total of 3 patients. That's a terrible sample size. 3 does not mean the entire population. So I'm like, and if I'm having a hard time trying to find, you know what I mean? How many patients? From all patients. We first detected one in one patient and then transmitted to other patients. Well, how many? I don't even see your figures or your table. See? I'm just scrolling forever. There's all your sources. That's great. Oh, here we go. Here we go. Finally. Patient 16. They had 16 patients. Here we go. Finally, the data. This is what I mean, guys. 16 people does not a, trend make. So they're going out and publishing all of this stuff. Now, this is useful, but the way that the media runs with this is they don't understand. 16 is not a huge number of people, So I would read through this and I'd be like, okay, that's nice, but that is not indicative of an entire population, and that's the problem I have a lot of with the science journalism. So it's like here, with stat going, people shed high levels of coronavirus. Study finds study of 16 people. But most are likely not infectious after. Of 16 people. 16. We have over a million. We have almost 2 million cases of coronavirus right now worldwide. Almost that. 16 people is not going to be representative of 2 million cases. You know what I mean? It's just, And that was, so that's the problem that I have with that. 16 people. Don't, it's just, whatever. It's fine. This is fine. I'm having a fit. Hello, Zalrat. I'll just say Zal. I'll just say Zal. I know you prefer that. Okay, let me catch up on the chat really quickly. Alrighty. Okay. I'm worried about my 25-year-old son, but with him driving to PEI and breaking a finger at work and being hospital, how long till I can let him in my home? Oh gosh. Yeah. This is probably at least 2 weeks before, after he self-isolates, at least 2 weeks. It's like science journalists. Media population. Media, yeah, that's the problem. Yeah. Oh gosh. Hey Johnny, how you doing? 16 people. That's on par with Wakefield studies. Exactly correct. Exactly correct. Zal, that rhymes with Hal. Got it. Hal 9000. Zal 9000. That's what I'm going to call you, Zal 9000. Oh my goodness. It's just like, what in the world? So let's have a look really quickly. So I showed you some of my sources. Here are the rest of them. I clicked on the links. The stat news, that made me nuts. Stat news. You know, I will read studies for you and tell you what they say, and if you want somebody else to write your stories. Man. And so, yeah, do check out NCCBI. Oh, I listed that source twice. I showed you the NIH thing on immunology. You can check that out. There's also some easier to read things. Let me show you some easier to read stuff. If you just kind of want the rundown on things, this was a pretty nice website, pretty straightforward, from Michigan Medicine. You can click on and get a bit more information. Here, let me blow this up. You can't see that so great. So this was pretty nice. It's just standard health information. I found it to be a pretty good website. Michigan Medicine is pretty good. Straightforward stuff, really good. Why it's done. It's good, straightforward thing. Here's another pretty neat site. Talks a bit more about PCR. If you want to get into more information about ELISA, skin biopsies, electron microscopy, resistance testing, that sort of thing. If there's any more, it's pretty straightforward and seems to be pretty good. So I thought that was a pretty good source. And then, yeah, there was the NIH, NCBI, Immune Defense, Chapter 50 from Medical Microbiology, 4th edition. We try to be legit here at the Science of to help you with our sources. All right. Level of no is at 9,000. Okay. So here we go. Also, here's where you can find me. ScientistMel.com. I've had some website issues, but I also have a shop there. If you haven't checked out my shop, you can check out my stuff right down there. Here's my store commercial. Heads up to Peter. Listen, I'm going to go for it for that. He has a tribute that came out for me recently. I guess I could probably show that. It feels weird showing a tribute on my own channel. I feel like that's just a bit kind of strange. But, you know, do check it out. Shout out to Peter for that. And so, yeah, you know, that has been the science of viral testing. I stream Saturdays at 10 a.m. Central. Sometimes it's an hour. Sometimes it's about an hour and a half. I usually try to wrap it up around about an hour. But sometimes an hour and a half is also important. Let's see. Goodness. I hope you guys are okay. Oh, mistaken reaper. You're immune compromised. You can't be vaccinated. It's been 11 years of my life in the hospital. I'm so sorry to hear that, Mistaken Reaper. I am staying home for you, buddy. I'm staying home for you. I'm staying home for my sister. I'm staying home for my mom. I'm staying home for all of the individuals who have who are a high risk population. Um, I'm at I'm staying home for you guys, and I'm trying my best to spread the word about staying home if you can. Also, Nina's tired. She's like, "This has been a great episode. I'm checked out now." So, she's like, "I'm checked out now. No more science. I have scienced enough today." So, um, next week, thank you, Jen. Next week, I guess I'll probably update everybody on news. I've been doing updates about coronavirus, the COVID-19. I've been doing updates talking about testing. I'm trying to maintain some positivity as well, but that, it's hard to do in times like this. But yeah, do check out Hannibal and Monty's show at the end of this month. I will be talking about the Tiger King. So if you've not seen that, go watch that train wreck. And so I'll be going through that with Hannibal and Monty, reviewing the Tiger King. That's part of their torture episode. Oh my gosh, so that's interesting. And do check out their show. Nods to Omnieris and Doc Savage for sending people over here to me. Thank you, Doc Savage, for your information in regarding epidemiology and for keeping me updated. One of my good friends in Omnieris. Go check out their channel. I was on Pimp Monk's channel yesterday. Check him out with Missy Ann. And I'm going to read from Mr. Rogers. And then we're going to let it go. All right. All right, here we go. I just opened this up. This is the world according to Mr. Rogers. I miss him so much. All right, and this is the courage to be yourself. This is from the section Courage to Be Yourself. When I was a boy, I used to think that strong meant having big muscles, great physical power. But the longer I live, the more I realize that real strength has much more to do with what is not seen. Real strength has to do with helping others. So in this case, real strength has to do with staying home, washing your hands, and looking out for each other in this particular virus. So there you go. So thank you for being here. I'm hoping to bring back This Week in BS soon. It's a bit hectic. Also check out Taste of Science, tasteofscience.org. I will be helping them, and I have been helping them coordinate their festival also at the end of April, prior to Hannibal and Monty's show. So watch tasteofscience.org. I'm one of their event coordinators, and we will be live streaming all kinds of sciencey goodness from their channel. Check it out. And I'll end with this. You always make each day special for me. You know how? By just your being you. I will see you guys next time. Sending squishy science hugs. Nina's tired, but she says, bye.